in two very common species that are Scyliorhinus stellaris and Scyliorhinus canicula. So these two sharks are morphologically similar. The main difference stands being the size, since the stellaris is known to get larger sizes. But focusing on the target species of this project, so Scyliorhinus canicula, this species is a demersal elasmobranch that inhabits waters, temperate waters, specifically the northeastern Atlantic Ocean and the Mediterranean Sea. Here in the Mediterranean, it is known to reach a maximum length that is comprised between 55 to 65 centimeters, and it can be found on sandy or muddy bottoms in a quite wide depth range, spanning from a few meters to almost 800 meters, while it can be commonly found between 100 to 300 to 400 meters. And since we are talking about catshark, all the species in this group are known to be oviparous species. So the reproduction takes place through the production of an egg case that is usually anchored to solid structures on the sea bottom, such as macroinvertebrates or macroalgae or rocky formations in general. And the small-spotted catshark is also known to have a quite wide reproductive period that, for example, for the French Mediterranean waters, goes almost all year round, with the exception of some months after the ending of the summer season. Being very common in these waters, in the French Mediterranean waters, this species is very commonly caught by commercial trawlers, and mostly as bycatch and also in a minor part for consumption purposes. And it is interesting because different studies have highlighted how under the right conditions, when this species is caught by bycatch and it is then released into the water, it can have a high survival rate that can get up to 90%, almost 90%. But still, also given this information, the exploitation status for this species in the area is quite poorly understood. So for this reason, the objectives of this project were to use the MEDITS data from that started in 1994, and it is still going on, to try to first investigate the life history parameters for this shark, and also try to analyze the spatio-temporal distribution in the Gulf of Lion. That was the area that was interested in the project. And lastly, to try to assess the status of the population here. When we talk about MEDITS, we refer to the Mediterranean International Trawl Surveys. That is a scientific trawling campaign that started in 1994, and it is still being carried out in the Mediterranean Sea, obviously. And each year in this scientific campaign, fixed stations abroad the Mediterranean Sea are trawled in the months between May to July, so between the ending part of the spring and the beginning of the summer. And the depth range that is interested for the trawls, the scientific trawls, is very wide. So we go from 10 meters to 800 meters, and the standardized gear permits to confront different trawls from different areas, from different periods. And the main outputs for these scientific surveys are density data, biomass data, length, weight, and also sexual and maturity information. To simplify the exposition of the results of this study, I divided the results in three different parts. So first, I'm going to talk about the life history estimates for the small-spotted catshark in the Gulf of Lion. Then I'm going to focus on the spatio-temporal distribution, and lastly, on the stock assessment part. So to begin with the life history estimates, for this part, I've been working mostly with the land frequency information for the species. And here, for example, we can see the aggregated land frequency distribution of the species in the Gulf of Lion for more than 30 years. So from the beginning of the MEDITS, 1994, to the last year, 2025. And we can see that starting from a few centimeters, like 6 to 8 centimeters, we get a gradual increase in density up to the size class of 40 to 50 centimeters, which is in the sample the most represented size class. And after 47 to 50 centimeters, it is also clear to see a sharp decline in density. So given that, the next step was to try to study the growth of this species. And the first assumption was to assume that the small-spotted catshark grew following the von Bertalanffy growth function that is reported here in the image. And this function describes a growth curve, that is the curve that we see in the same image. And this growth curve describes the growth, obviously, of an organism over time, where the growth is expressed as length on the y-axis and the time or age, usually in years, is on the y-axis. And this curve can be described by three main parameters. The first one is the asymptotic length, or the theoretical maximum length that a species can reach, assuming that the organism is able to live for a very long time. In the second place, we have the K parameter, or the growth coefficient, that graphically can be visualized as the curvature of the growth curve, and it describes how fast the animal is growing over time. And lastly, we have the T0 parameter, that by itself, it doesn't really have a meaning, but the purpose of this parameter is to anchor the growth curve to the age axis in the way to get an estimate of the length at time zero, that is the time at birth. And for example, for this shark, the observation say that at birth, the species should be around 6 to 9 centimeters. So if we talk about bony fishes, where we can estimate the age through readings of the otoliths or other bony structures, it is possible to build this curve directly, just measuring and aging the fish. But since we are talking about a shark, and for elasmobranchs, the story is a little bit different, since it's much more difficult to age directly these animals. I had to go through the analysis of the length frequency data. And to explain how it is possible, I wanted just to introduce how length frequency distribution can be used. So here on the left, for example, we see a perfect length frequency distribution example, where we have a peak in abundances, in densities that can be theoretically divided in different age groups or different cohorts. So for example, we can see the first peak, that is age 0, then we have 1 year old individuals, 2 years old individuals, and so on. And the idea is that the density tends to decrease over time due to natural mortality or also fishing mortality if it is present. So in this way, it should be possible also visually to separate the different age groups or the cohorts, and having the possibility to work with different years, length frequency distribution from different years. It is also possible to build something like what is reported on the right part of the slide. So here we see the same distribution, but different years. We are going to obviously have different distributions. And with the von Bertalanffy growth curve, it should be possible to try to track the evolution of the cohorts through time. And this is the theory behind what I did. So here, leaving the example behind, we have the actual data from the MEDITS trawls. So each horizontal line represents a year. We have more than 20 years, so we go from 1999 to 2025, and the black polygons represent density peaks. To do this analysis, I use the ELEFAN program, that stands for Electronic Length Frequency Analysis. And briefly, it does what I just said. So it tries to fit different curves with different sets of parameters. And then it just selects the best fitting curve, so the curve that passes through the most amount of positive density peaks. I applied the ELEFAN for males and females separately, and this is the final result. So I got two different growth curves, one for the females and one for the males. So the parameters, they look really similar. So for example, we have the asymptotic length for the females, that is 61.6. and the K parameters 0.22, and for the males, respectively, 62 and 0.25. So just looking at the numbers, they could like very similar. But if we look, for example, at the age of maturity, which I'm going to talk just in a few minutes, we can see that there is just 1 centimeter in difference, but it's translated in years. That is possible using the inverse function. We have almost 1 year of difference. And so the more we go towards larger sizes versus towards older individuals, the more we can see the difference between growth. Before moving to the last plot for this first section, I wanted to say that in the next parts, I'm going to talk about juveniles. And when I'm going to talk about juveniles, I'm going to refer to the individuals that are smaller than 20 centimeters, since these individuals are the young of the year or the individuals that are born in the first year of the sample. Then, for the last part, the reproduction part, in the MEDITS data, we also had maturity information. So using this maturity information, it was possible to build a maturity ogive and to estimate the L50, which is a parameter that stands for the length at which 50% of the individuals can be considered sexually mature, and is usually used for just to assess when which size an animal reaches a sexual maturity. And as we can see, we have a slightly lower maturity size for males compared to the females. And as I was saying before, we have 1 year of difference. So the males tend to get to sexual maturity at smaller sizes and 1 year before the females. But more interesting is the sex ratio that here I estimated as the proportion of females for each length class. So the red dotted line represents the equilibrium status. So when the values are around the dotted line, it means that we have the same proportion of males and females. And we can see that up to 40 centimeters in size, the proportion are more or less in equilibrium, but after 40 centimeters, that is around the age where these animals reach sexual maturity, we see a drop in the sex ratio. So this means that after this size, in the large individuals, we have more males than females. And there are a few theories to explain this pattern. One is that since we are talking about oviparous sharks, the energy investment for the females is much larger compared to the male investment. And this could mean that the females, after they reach maturity, they tend to have a lower growth rate compared to males, and this could also mean a higher mortality rate. So this could explain why in the large individuals we just see more males compared to the females. So this was it for the first part. Now I'm moving to the second section of the results that is related to the spatio-temporal analysis of the small spotted catshark in the Gulf of Lion. So here we see two different plots. We have here I worked not with the length frequency data, I worked mostly with density that is standardized and it is expressed as number of individuals per square kilometers. And on the left, we see an interpolation of the density values in the Gulf of Lion. And on the right, the same thing, but so density, but plotted on a depth gradient. And in both of these plots, we can see that density tends to be quite low in the first 100 meter, in the first part of the continental shelf, while it peaks between 100 to 200 meters with the different density hotspots. And then it tends to decrease over towards greater depths. So we have the higher density in the lower part of the continental shelf and in the upper slope. I also tried to look for any pattern of spatial segregation by sex, so dividing males and females, but at least for the spring period where the MEDITS trawls are being carried out, we didn't observe any particular segregation pattern. So after that, I moved to looking at any differences between different sizes. So as I was saying before, here we see the difference between juveniles, subadults that are between 20 to 40 centimeters. And after 40 centimeters, as I said before, that is the length at which they reach sexual maturity, we can consider them to be adults. And it is clear that the juveniles are located in two different hotspots, density hotspots in the Gulf of Lion, one on the western part and one in the eastern part, while the subadults and more clear for the adults, they are more evenly spread across the area. So they are more present in the central part of the Gulf, and more importantly, they are also more present in the shallow waters where we don't get the juveniles. So given the peculiar distribution of the juveniles, I tried to look if these two hotspots were consistent over time, and they actually were. So for more than 30 years, we can see that these two density hotspots are present between 100 to 300 meters. And looking at some definitions for nursery, for sharks, we see, for example, three requirements. So the juveniles must be more common in these areas. They have to have the tendency to remain in those areas, and those areas needs to be used repeatedly across the years. And given these three requirements for what we have seen, it seems reasonable to think about these two density hotspots as potential nursery areas for Scyliorhinus canicula in the Gulf of Lion. The same pattern was also observed in other areas of the Mediterranean. It is also possible that more nurseries are present. We just don't see them given the trawls of the MEDITS. And for the last part, the stock assessment part, when we talk about the stock assessment, we refer to a process which is the purpose of evaluating the status and the dynamics of a population in a defined area. And the main objective of this process is to simplify the management of the stock or the portion of the population in the area. Since I've been working with a really good data set of length frequency data, I decided to use two different models that are both length-based models. The first one is the LBI or length-based indicators, and the second one is LBSPR or length-based spawning potential ratio. The first one is quite a simple model. It was developed for data limited or data poor species in the first year of the 2000. And I say it is simple because as an input data, it just requires length frequency information that in our case, in this case, I selected just the last 5 years from the MEDITS data. So between 2021 and 2025 to get an overall picture of the most recent years in the area. And optionally, we can also use the asymptotic length that we have estimated using the von Bertalanffy growth curve and the L50 estimated through the maturity ogive. So having all this information, the model gives as an output 3 parameters: the proportion of mature individuals in the catch, so how many individuals mature after reaching sexual maturity are present in the catch, the proportion of optimally sized individuals. This is a size that is after maturity and optimizes the catch. And in our case, it's very close to the maturity size, so we can consider them almost the same. And the most important parameter, in my opinion, here is the proportion of megaspawners. Megaspawners that are defined as individuals that are 10% bigger than the optimally sized individuals or the mature individuals. Applying this model to both sexes, males and females, this was the output. So here we can see the length frequency distribution for males on the left and females on the right, the 3 different size categories. And just looking at the numbers, we see that in general, the proportion of mature individuals and optimally sized individuals is higher for the males. But most interesting is the proportion of megaspawners. So usually the target is set at 20%, but for the males, we have a proportion of 6.5% that is really below the target, but for the females, we just get 0.1%. So it means that there is a clear scarcity of very large females in the sample. And after looking at this information, I tried to look to use the other model, the LBSPR, to try to confront the results. So this is still another length-based models, is a bit more complex than the LBI since it needs more input requirements And still, I had all this information. I also estimated the natural mortality, which I didn't specify before, but I have the value for both females and males. And in this case in particular, since we are talking about the SPR, the spawning potential ratio, we are referring to the spawning biomass. So this model was applied just to the female portion of the population. As an output, we get 4 different parameters, but the 2 most important ones are the F on M, then stands for fishing mortality on natural mortality. The fishing mortality is the mortality that is caused by the fishing action, while the natural mortality is more or less everything else that is not related. And once we get an estimate for this ratio, knowing the natural mortality, it is possible to also get the estimate for just the fishing mortality. And the SPR, or spawning potential ratio, is the real reference point given by this model. And it represents the observed spawning biomass, so the biomass of females that we observe in this moment, compared to the virgin spawning biomass, so the spawning biomass in an unfished scenario. So applying this model just the same period, so 2021, 2025, to the MEDITS data, the estimates that I got were these ones. So 6.1 for the ratio and 9% for the SPR. This means that the fishing mortality was estimated at 2.1, that is kind of a really high value. So a really high level of fishing mortality, and the SPR is 9%, while the target is usually set at 40%. So 40%, it means that the population that we are observing is at 40% compared to the original population in a non-fished scenario, while the limit that should not be exceeded is 20%. So what we see here is that we are way below this 20% limit. And so the scenario given by the model is it's not the best one, but the reason to try to explain these results is most likely related to the sharp decline in density above the maturity level that we can observe here. And we also observed in the overall aggregated land frequency distribution for the 30 years. So the only way the model has to understand, to interpret this sharp decline in density, is to assume a really high value of fishing mortality and a low value of SPR. So what the model does with this kind of shape for the population is probably overestimate the fishing mortality and underestimate the SPR. So there could be different reasons, but all for this shape of the population. But still, it's surely needed more work to try to understand why there is this decline after reaching the adult size in the population. This is another plot given by the model. So in the light blue or green shape, we see the actual shape of our population in the last 5 years, the female population. And in red, we see what the model thinks it's the best theoretical fit for the population, assuming an SPR of 40%. So we see that in our population, we don't get a lot of subadults. And this is, we can also try to understand why we don't get this kind of portion of medium-sized individuals. But most importantly, we see that we don't have the large individuals, at least in the females. Or maybe it's more visible in the females than in the male portion of the population. So to resume everything all together, in this project, we have been seeing 3 main points. So the first one is that the possibility, well, more than the possibility, the actual different growth rates between females and males. And this difference leads to a different amount of individuals between the large size individuals. So after 40 centimeters, as I was saying before, we have more males and not a lot of females. And this could be related to the reproductive strategy or other reasons. While for the spatial distribution, we have seen that we have a spatial segregation. So we have 2 nursery areas where we found the juveniles on the slope, on the upper slope, or on the lower part of the continental shelf, while the subadults and the adults tend to spread more evenly across the area. And lastly, the pattern that we have observed through all this process, so the lack of very large individuals, in particular in the female portion of the population, has a really strong influence in the model results. And so there is still surely work to do on this. One possibility is that, for example, this species, the growth of this species does not follow the von Bertalanffy function. So maybe other function can be describing best the growth of the species. Or maybe one other possibility is that actually the fishing mortality, the fishing action carried out through time could surely has an effect on the presence, on the fishing mortality for the larger size females or males also. Or another possibility is that we just don't get to see the large females in the Gulf of Lion. But still, this last one, in my opinion, it does not seem very likely, since different studies have highlighted how this species is not known to have a wide range of motion with a radius of 30 kilometers per year maximum. Well, not maximum, maximum can be higher, but since it's a demersal elasmobranch, it tends not to move too much. So in 30 plus years, it is odd not to see any very large individuals in the area, since also the MEDITS trawlings, they cover quite a wide area here. So there could be different reasons behind the shape of the population and the influence that this shape has on the results. And surely more studies are needed, more information are needed, both on the growth of the species, both on the distribution of the species, and also to try to use maybe different stock assessment models that fit best in a best way the characteristics of the species itself. And with it, I finished. Thank you.
Thank you, Daniele. Do you have questions?
What did you estimate your breeding mortality rate? And if it have been done with your data set, maybe you can assess that rate according to the sex or the age, and maybe it could explain why you have that decreasing of large female in your population.
You mean the survival rate after?
Yeah.
No, I did not estimate by myself. It was reported in different studies where they use tag and recapture studies to estimate the mortality.
Okay.
So I didn't find any information about the difference in mortality between females and males, but it could be also related to that, since this species is caught mostly as bycatch. So the juveniles or the young individuals, the small individuals tend to be released, while the large individuals, sometimes they are kept for commercial purposes to be sold in the fish markets. So it is possible that the fishing mortality just is higher for the very large individuals and can affect also the distribution, yes.
Okay. And did you have any information about some learning behavior? The more you catch a specimen, the less is catch. Like you have some species that act like that.
Sorry, what? Like,
you know, if you have any, that species maybe have a behavior, a learning behavior that they are not catch. The more they are catch, the less they are catch. I don't know if you understand that.
Yes, no, I didn't find anything regarding this part, no.
Okay.
Other questions?
Yeah, I wonder if the pattern you've seen on the large females is maybe just a byproduct of sexual segregations.
It could be.
I missed, sorry, I arrived a bit late and I missed the sampling effort, the spatial distribution of the sampling effort.
Yes, so.
Because I know that on this species, you have some difference in male and female behavior, and they try to avoid each other at some point.
Mm-hmm.
I wonder if it could be.
Yes, so here what we see in the map are the MEDITS trawls in the Gulf of Lion. So the coverage is pretty evenly spread, I would say. And for what regards the spatial segregation, I showed a plot before where they look at interpolation of density, and for the MEDITS period, at least, sorry, here, both males and females seem to be distributed quite evenly across the area, but some studies from some areas, in particular in the Atlantic Ocean, report the presence of a sexual segregation where females tend to avoid the males because of the harassing that follows the reproductive action. But for some other areas in the Mediterranean, like for example in the Aegean Sea, no sexual segregation was found. So it is
the adult ones or it's all combined?
all combined here.
Maybe you could try to look at the different stages.
Between sizes, yes.
See if, maybe like the larger females just disappeared because they use a different habitats and you don't see in your samples.
Sure.
But it's quite curious that, there is such a drop in the female ratio. They should be bigger than the males.
In this species, not. It has been observed that it's for the species it's normal to have larger males compared to the females. But it's odd to see this kind of sharp drop just a few centimeters after the maturity size, yes. But it's a possibility that the segregation by sex for these causes can affect the low presence of females, yes.
Yeah, thank you very much. It was really nice. I was wondering if I remember well, in 2020, the campaign was valid for several months. And I was wondering if you looked at that specific year, whether the juvenile hotspot maybe moved it around or not at all.
I didn't look specifically at year by year.
And so that in 2020, we did the survey in only 3 weeks, and it was in October. In October, it's bad weather, so the juvenile hotspot, they haven't been sampled at all because we couldn't go there. So we cannot answer.
Sad. Nice.
Yeah, other question.
I want data from the fisheries actually. I mean, do we get any data on the species?
Yes.
And stuff?
I didn't include the commercial data for time reasons. But here, for example, the LBSPR, I also applied it to the commercial data from the last 4 years, from 2021 and 2024. And we see that the shape of the length frequency distribution from the commercial data is different. So we still have the only actually size class that is represented are the adults, while we lack the presence of juveniles and subadults. And well, this can be explained simply by the difference in the mesh size. So the MEDITS mesh size is 20 millimeters because the purpose of the MEDITS is to catch also the juvenile portion. And also, since we have seen that there is the possibility of the presence of nurseries in the upper slope in areas that are less accessible to the trawling action, the commercial trawlers probably they just cover the shallower waters where we have seen that we just have the adults and subadults. So this could explain this pattern here for the commercial data.
Not to mention the discard. No, this is OPSMER.
This is the discard plus the landings altogether, yes.
Okay.
And where are the females and men? I mean, do you see the same drop in the females? The larger females are disappearing also?
Yeah, this is just the females. So the drop we see is the same that we observed in the MEDITS data. So after like around a little bit higher than 40 centimeters, then there is this sharp drop in abundance also for the commercial data.
That's nice. That's also what's different from the MEDIT data. I mean, it's always at the same time for a few weeks. I mean, there you're looking at what's happening over the year. That also answered Joanne's questions. Yeah, that we have a segregation.
Thank you, Daniele. So you said you couldn't use age data to create your growth curve, but could you get any data to validate the like the curve you end up having, or to compare estimates with other studies?
Yes, I compared it with some other von Bertalanffy growth curves that been estimating in the Mediterranean, and usually the estimates are a bit lower. So there are not a lot of studies in the Mediterranean. There are more on the Atlantic Ocean, and in the Atlantic, actually, they also try to read the vertebrae. So they had a more precise, I would say, readings. And like, for example, they estimated a longevity between 12 to 13 years for the species in the Atlantic. But still, we know that there is a difference in size and probably also in longevity between these two areas. Since in the Atlantic Ocean, the reports say that Scyliorhinus can grow up to 80 centimeters, while in the Mediterranean, it was never observed reaching this size. It's smaller. And so our estimations are quite in line with the other Mediterranean estimations, a bit higher, but I think it's also because we have more years. Usually the other studies, they just try to fit the curve using length data from 1, 2, or 3 years, while we had more than 20 years, and we also had the opportunity to observe larger individuals. So that could be why we have slightly higher estimates. So, for example, the estimates for females and males in our case, in this case, were around 61 centimeters, 62 centimeters. And in other studies were around 55 or 48. That seems quite low since we have observed that they tend to grow larger than that.
Okay. And another question. I don't remember. Yes, okay, sorry. You say you have 20 years of data with the MEDITS.
Yes.
Do you have any, because you've been analyzing kind of the length frequencies, do you have anything related CPUE or something, like how these vary over time? Is it different? Is it decreasing?
I don't have the CPUE because in the MEDITS usually it's the density or the biomass are standardized. So we use these biomass indexes or the density indexes. I didn't have time to estimate the CPUE by the commercial data to look into it. So I just look at the density and also the biomass that I didn't include here. But the overall pattern of the biomass was similar to what we have seen with density. So a peak in the lower part of the continental shelf. And sorry, I just wanted to go to the density. So here, the biomass was following a similar pattern. So peak on the lower part of the continental shelf and then a decrease in biomass moving on to the greater depths.
And there is no signal regarding temporal trend? Like,
no, I looked into the, I tried to do some box plots to look at the length, for example, over time, but it's quite variable. It's not visible a clear trend over the 30 years, no.
Thank you.
I also have a question. Do you aim to translate your work into management measures?
I think it should be possible, but not with the results of these models because they are too influenced by the shape of the length curves, and there's some assumptions of the model are not satisfied. So I think it would be interesting to try different models, maybe models that don't work with length-based measurements, but maybe with biomass or other kind of models to try to look if the results are similar, if they give some more reliable responses.
Maybe with the nursery, so grounds.
Also, yes.
So is it the first time they have been identified in the Gulf of Lion?
Nurseries, yes. We have identification in the Tyrrhenian Sea, in the Ionian Sea, in the Aegean Sea, and in the northern part of the Tunisian coast, but here I didn't find any report of this.
And is there a specific characteristic for those like spots?
Or here?
Yes, like apart from being spots of higher density, do they have like specific characteristics which could?
Well, the temperature was very similar, so to the areas, neighboring areas. So It can be influenced by environmental variables. It could also be influenced by trophic reasons. But for example, in the Atlantic Ocean, it was seen that the spawning action was happening in the shallow waters. So it's quite a contrast between here that we have deep nursery areas, and in other areas we have the spawning grounds in shallower waters. So one idea is that the stable environmental conditions that are specific, maybe characteristics of the Mediterranean Sea could help these species to spawn in deeper grounds where the conditions are more stable than in other areas compared to the Atlantic or compared to the shallower waters. But still, please.
You have looked around Corsica?
No.
Because I think in Corsica you have some, you can find some eggs like in shallow waters.
Yeah.
30 meters.
It's here too. The MEDITS survey is not well suited for very coastal coverage. So we go up to maybe 20 meter depth. No, that's the shallower we go. So we have identified these two deep hotspots. Doesn't mean that there is no. Maybe there are others, but we don't see in the coastal area.
But yes, the reports are contrasting. In some areas, they appear to spawn in very shallow waters. In other areas, they appear to spawn in deep waters. And it will be interesting to try to look why there is this difference between areas, if it's related to environmental variables or trophic reasons or whatever, any other reason, yes.
Do you agree with the temporal trend of the L50 or?
No, I didn't look at the temporal trend of L50. I just use all the data together. Something that could be interesting to look at, yes.
Other questions? Maybe online? No. So, thank you.
Thanks everyone for coming on a Tuesday, and see you on Thursday for a presentation on the DAL.
But it will be in Montpellier.
It could be in Montpellier, but we'll open the place here as well. Thank you.
Warm up for you.